Hello, my name is Laura Zanko, and I'm an early-stage researcher at Biotalentum. I work as a doctoral candidate inside the Medicine Made to Measure, or MMM project, with my mentor Dr. Balog and supervisor Professor Dinyash. Most of us have personal relationships and encounters with people affected by rare neurodegenerative diseases, be it multiple sclerosis, ALS, cerebral palsy, or Huntington's disease. They are characterized by a wide array of symptoms like confusion, memory loss, behavioral changes, pain, coordination issues, and fatigue. Many are quite unknown to the general public and even scientists and medical professionals. In this group of disorders, there are many which affect maximum 30 individuals worldwide, with each of them presenting unique genotypes with varying mutations between patients, thus making them not ideal candidates for potential treatment development. These are so-called non-rare neurodegenerative disorders, which are the focus of the MMM project, one of them being ataxia telangiectasia. On one side, pharmaceutical companies do not see any commercial profit from treating these patients, and on the other hand, their unique mutations could hinder the development of an efficient therapy method. All of these factors contributed to the rise of individualized therapies for rare diseases, with antisense oligonucleotides at the front of innovation. ASOs are short synthetic strands of nucleic acids designed to bind to specific RNA molecules produced by faulty genes. They modulate gene expression, thus correcting or bypassing the effects of mutations. Concerning their mechanism of action, after binding to the corresponding RNA, ASOs can promote the degradation of messenger RNA or inhibit translation to hinder the production of mutant proteins, or modify splicing to create a functional but truncated protein. ASOs have been approved for use in rhinitis induced by CMV, familial hypercholesterolemia, and other genetic disorders. Concerning rare neurodegenerative disorders, there are multiple ASOs approved for Duchenne muscular dystrophy, one for spinal muscular atrophy, with the newest addition being tofersen for amyotrophic lateral sclerosis. There is ongoing research for ASO therapy in Huntington's disease and many other disorders. ASOs present advantages as they can be customized to target specific mutations and genes, which should not only alleviate the symptoms as currently available therapies do, but affect the actual cause of the disease. Challenges are present as we need to ensure the ASOs are delivered to the appropriate tissues, and we need to carefully evaluate the long-term safety profiles to avoid off-target effects and unfavorable immune responses. ASO therapy represents a promising approach for treating rare diseases by directly targeting the genetic causes of these conditions. The MMM project aims to establish an efficient, safe, and novel ASO therapy model for patients with ultra-rare neurodegenerative diseases. In the drug design and development pipeline, we started by identifying the genetic makeup of the patient to find the specific mutations related to the disease. Based on that, the drug can be designed and synthesized. After producing the drug on a small scale, its efficiency is tested in relevant disease models, continuing with toxicology studies, while statistical analysis, evaluation of patients' response profiles, and ethical evaluation are done before starting trials in humans. During the preclinical stage, a vital component are toxicology studies, aimed at assessing the safety of a new drug candidate before it is tested in humans. These studies help identify potential toxic effects and determine safe dosage levels. The results are essential in the design of clinical trials and regulatory submissions. Key aspects are identifying whether the drug has any harmful effects on biological systems, to establish the relationship between drug dosage and its toxic effects, which results in establishing safe dosage levels in order to minimize the risk of adverse effects in humans. Toxicology studies are done as in vitro studies in cell lines, while in vivo studies are done on animals. Our laboratory will be responsible for developing cytotoxicity assays to measure the toxic effects and determine the dose-response relationship of the ASOs using human-induced pluripotent stem cell-derived neuronal cultures.